Welcome to the webinar. My name is Kimberly Horndusky and I'm with the Economic Growth and Endangered Species Management Division at the Texas Comptroller's Office. Today we have Dr. Tim Bonner from Texas State University to discuss his findings from the final report on the prairie chub. If you'd like a copy of the final report, please contact me by email or phone, which is listed on your screen now. Without further ado, here's Dr. Bonner.
Thank you, Kimberly. Before we begin, I want to highlight the number of students and also Oswald, which was a visiting scholar, that took leadership role in this project, which means that they were paramount in the field as well as processing specimens in the lab. And of course, with my co-PI on this project, thanks to Nolan Martin. So the study area was the Upper Red River drainage, which is the Red River drainage of Texas, including the Oklahoma section of the Red River all the way to Lake Texoma. This was our study area because this is the breadth of where we have the prairie chub, which is our focal species for this project. And part of the reason why is prairie chub is listed as a species of greatest conservation need. However, there's several other species within the Upper Red River drainage that also are SGCN species, including the Red River shiner and Red River pupfish. Both these have large abundances in the Upper Red River, as well as distributions outside of the area. And then we also have three other species, the silver chub, the chub shiner, as well as the goldeye, that are also reported in this area, but they definitely found other in areas outside of this reach and in higher abundance, except maybe the goldeye, which is abundant in the Lake Texoma itself. Objectives of this project was to quantify current habitats, fish distribution, and habitat associations for all fishes, assess changes in fish communities through time, comparing what we find today versus the historical records, with some of those and some of the reaches going back as far as the 1930s. And then we wanted to assess habitat associations more at a univariate level for the prairie chub, life history parameters, reproduction and diets. However, since Red River shiners, we were catching them at the same time we were catching prairie chubs, as well as plains killifish. Since information is missing for all three of these species, it was an opportune time to quantify their life history parameters as well. So our study consisted of two levels. We wanted to look at the complete coverage of the community within the study area, and that included 20 reaches, 36 sites, both main stem and tributaries of the Red River, and we sampled these twice within the year, generally during the hot season and then in the cooler season or winter time. For the life history work that we did the community work first to find viable, sustaining populations of the prairie chub. So we started the prairie chub life history work a little bit later after we had an idea of how the fish were assembled in the Upper Red River. So we selected 3 sites on the Peace River for life history, and then 2 sites on the Wichita for monthly collections, starting right at the beginning of what we thought would be their reproductive season in February or March, and then it went for 1 year. For the community-wide sampling of the 36 sites, at each of the sites we sampled and quantified all available habitats that were available to us. The mesohabitat level, mesohabitats included riffles, runs, pools, and a few others. And then we did the traditional measurements and quantification of the habitats, the water quality measurements, depth, substrate types, veg type, current velocity. All fish were identified and enumerated by habitat. As we knew that Macrobopsis australis, the prairie chub, as well as the shoal chub, was found in the area. We used the keys provided by Eisenhower to split them out. However, with our genetic work, we did find that there were hybrids of the two, but we did not distinguish the hybrids in this study. We called them one or the other species about how well they fit the Eisenhower key. For life history, we visited 5 sites monthly, and our target number, we collected up to 5 female per species, and that's what we use for reproduction, other life history and diet assessments. And we also, while we were there, quantified the habitats that they came from. For results of the community, we captured over 36,000 fish, which represented 13 families, 49 species, and sampled 400 mesohabitats. As far as the overall look at the community among some of our more abundant species, red shiner was the dominant form, followed by the Red River shiner, plains minnow, and then gambusia. If we look at our SGCN species, the Red River shiner, Red River pupfish, and the prairie chub were among the more abundant species within the drainage. We did not capture the chub shiner, and a few of the silver chub as well as the goldeye were captured near Lake Texoma. We used PCA to describe the 400 mesohabitats along a gradient of mesohabitat type, current velocity versus slow-moving waters and pools. So majority of our samples were taken in among the mesohabitats were swift water runs with sand substrates. As you move deposit scores of PC1 is explaining most of the variation in these 400 mesohabitats. We have slower pools on this side with the silt organic type substrates. If we take an average PC1 and PC2 scores together for a site or for a river reach, we have the 20 reaches listed here. What the gradients that this shows us is that towards negative PC1 scores towards this size was predominantly our prairie stream type habitats. And those were located, it's also showing a west to east gradient as you move further east towards Lake Texoma. The tributaries then became a little less stream type towards Lake Texoma. Here's an example of what we think of as a prairie stream habitat characterized by broad floodplain, broad channel. Oftentimes the channels can be braided, shallow, tend to be a little bit higher in conductivity, and maybe even higher in water temperature. And then as you move towards the eastern, definitely the tributaries along the eastern reach, then it became more entrenched channels and more of that traditional run, pool, riffle type substrates, or run, riffle, pool type habitats. So we use CCA to link the 400 habitats and the corresponding fish community all together. So the habitats parameters that help explain the fish habitat or fish associations. One of the strongest gradients came out was specific conductance or salinity of the water. So over here you tend to have more of the prairie stream, western type drainages and habitats. And then as you move this direction more to the larger lower reaches of the Red River upstream from Lake Texoma into some of the eastern tributaries. We look to see how the fish were associated. We have the 5 species that we think of mostly being prairie stream fishes, such as the prairie chub, plains minnow, Red River shiner, plains killifish, and the Red River pupfish tend to be more associated with these types of environmental gradients. Whereas our other, what we're calling among the abundance, greater than 1% in relative abundance, we've got a cluster here of fishes, emerald shiner, bullhead minnow, as well as the red shiner that tend to be associated more with the Red River mainstem proper, and then eventually a group of fishes that are traditionally found in more of these entrenched type tributary habitats, especially with the gambusia, where more of their abundance came from these types of habitat. If we look at all the fishes that are less than 1% in relative abundance. Most of all these were found in the lower reaches of the Red River main stem, as well as the tributaries, starting somewhere around the Little Wichita River and on downstream. Since specific conductance came out as one of the strongest gradient on CCA, we did plot the range of segregation, or the association with conductivity for more abundant species. And then if we look at this bottom panel, this is amount. These are the distribution of what was available with respect to specific conductance. Majority of our habitats were found in lower conductivity levels, even though lower being 10,000 microsiemens per centimeter is still fairly high salinity among freshwater environments. But then as you moved upstream, we had more of the higher conductivities. So we see variability with several of these species, wide ranging among the different conductivity levels. And then as we move towards those that are found lower in more eastern sections of our drainages, tend to have more of a narrow distribution with salinity. The high variability up here made somewhat sense. If once it rains, and it can greatly decrease specific conductivity in a short time period. So it can range from high conductivity, rain, low conductivity, where we to explain the amount of variation that we find among some of our top species here that had a central tendency more towards the higher conductivity levels. Not only did we see some segregation among the community, we're also seeing segregation with specific conductance on two of our two species within the Macrobopsis complex, the shoal chub and the prairie chub. The shoal chub tend to be found in the lower conductivities, lower than 10,000 microsiemens per centimeter. Prairie chub, we would find them in the main stem, lower reaches of the Rio Grande, closer to Lake Texoma. However, we would also find them distributed well upstream in the Red River Basin at higher conductivities. So we're seeing that might be a limit on where salinity limit on where we would find and where the high ostema of the shoal chub can go. As far as the historical trends, we were greatly benefited by the work that Gene Wild at Texas Tech did back in 1996 by compiling historical records for the basin. So we used his estimates among the various reaches, which represent the different lines for the prairie chub, Red River shiner, Red River pupfish, and then we added our relative abundance on our most recent collections at these sites. We're still in the middle of assessing the historical trends, but we do note that we did see some decreases, but we're not seeing any what we would call wide extirpation events happening in some of these basins or in some of the reaches. So that ends the community aspect of it. Now moving into the life history aspect of our work and showing some of the numbers that we have here as far as the numbers that went into our assessments. I'm only going to take time to highlight what we see with what we found with the prairie chub. Thinking about redundancy within the basin, we found prairie chubs in 30% of our 20 reaches. Found at 44% of the sites and 13% of the meso habitats that were sampled. So going back to our PCA of the 400 meso habitats, we plotted where we found. So for each of these meso habitats where we found a fish, we colored it gray, and then we increased the size of the circle depending on if there were a few found all the way to a lot were found. And this gives us the chance to look and assess on how ubiquitous they're found in the amount of available habitats. And so they were spread out between the runs and in swifter waters, and then also found in some of the slower type waters. When we assess this by reach, so now we've got the 20 reaches, and highlight where we found them in these reaches. The reaches that we found them in, again, were towards the western portion of our drainages, tend to be the prairie stream type habitats over here, but they were not found in some of the prairie stream habitats, and these are the areas that tend to have the highest conductivities. And there were a few that had similar habitats, some of the smaller tributaries, but we were not finding the prairie chub. So there's a couple hidden in this cluster here. From a univariate approach, we assess the occurrence, so if they are present or not, if the prairie chub was present or not, and their abundance. So we compared occurrence and abundance represented in the bars, where they occurred versus what was available is represented, what is represented by the dark line. So if we look among current velocity gradients, if we looked at just occurrence, they seem to be found. They were found in, they occurred in about the same proportion as the habitats with the current velocities that were available. If we base it on abundance, we might see a slight tendency towards some of, slower type water, slower type current velocities. If we look at depth, both occurrence and abundance are suggesting that they're not in the shallowest water, but in moderate depths. And then as the water gets deeper, as far as they occur, they seem to be in about the same abundance as what was available, maybe a little less so in abundance. And also with specific conductance, both of these showing that they tend to be found in more of the moderate levels of conductivity, not in the freshest of water, nor in the highest levels of conductivity. Going out each site and measuring the fish by month, we're able to construct length frequency histograms and using a PHI-STAT program to help guide us in distinguishing which ones are age 0, age 1, and age 2 fish. This allows us to understand, say, longevity that lived about age 2, majority that in the reproductive season is coming from age 1 fish, I'll show next. And then we see our first occurrence when we were catching the age 0 fish in our seines. So from this, we can also generate growth rates of different age groups. And then also know among our sites, are they dominated by age 2 fish, age 0 fish? As far as the reproductive season, which we define as the first time that we see investment in energy into their gonads, that began in March and extended through September. Spawning season, based upon elevated GSIs, gonadosomatic index, as well as the ovarian stage, maybe as early as April, that they're probably prepared to reproduce. And then it looks like the spawning season was between April and September. And then the white represents immature or resting ovaries. So they exhausted their reproduction for that year, starting in October. We assess the diets. Primarily, they're consuming a variety of aquatic insects, but also will consume some terrestrial forms. High occurrence of sand in their diet suggests that they're a benthic feeder, but with their subterminal mouth, barbels, that corresponds, it makes sense. We're also set up to look at in-stream movement in the Peace River, as well as the Wichita River. The Peace River, the upper reach was, this panel represents the upper reach. This panel represents the lower reach, which are separated by about 100 kilometers. And so we wanted to see, there's thoughts that these fish are moving substantial distances upstream and downstream. So we're looking for signatures in that based upon which age groups that we find at each site. We're still in the middle of pulling all of this together. But what we did find is that we didn't have any of the prairie chubs at our uppermost site whenever flows were low, and at conductivities where we have not caught any prairie chubs. Once rains came, flows increased, in the area. Conductivity drop. We did start seeing age 1 and age 2 fish found at our uppermost site. So we're thinking about, are they moving for reproductive season? Are they moving for some other factor? These are some of the questions that we can address, hopefully, with this type of analysis that is still ongoing. But not only are we interested in just Australis and in the upper Red River, we're also interested in the whole Macrobopsis complex in Texas, because most of these, except Hyostoma, are an SGCN species, and they're all indicative of or indicator species of a flowing system and perhaps even a healthy system. When we have them, that is an indication that things are good, at least for the fluvial type specialists, except for Marconus, which may be more of a spring associated type fish. So this does fill in a big gap on just general life history, longitudinal gradients and reproductive season, a lot more about what we know about this Macrobopsis complex. We, again, I'm not going into the details, but we have similar information that we just reported for prairie chubs, for the plain killifish, and also for the Red River shiner. So just some few thoughts on this. We're still working on moving these to publications, thinking about what this all means and synthesize into the existing knowledge. But some highlights in the upper Red River drainage we found dominated by small-bodied, short-lived fish. Probably many of those are broadcast spawning, but not all. The significance of this is what we find dominant in the upper Red River drainage is pretty consistent with what we find in upper reaches of most prairie streams, especially in Texas and the Brazos, Canadian River, New Mexico, and the upper reaches and the Prairie Stream reach, the Pecos, and then even further north from here, consistent with what we find in other prairie stream habitats. As far as historical trends, so far in our analysis, we're not indicating any extirpation events. We do find that say for the prairie chub, it can be variable. So sometimes you can find them in a stretch, and then sometimes you can't. And that one possible explanation for that is the conductivity gradients. We noticed in the Prairie Dog Town Fort, which is in the Red River of Texas before it hits officially the Red River in New Mexico, is that when the conductivity levels were low, we've had a rain event, flows were happening, high flows were or moderate flows happening, we would find the prairie chub. We go back out during the warm season, flows are low, conductivities are high, we did not find the prairie chub. So they're moving perhaps back and forth, creating an appearance of, haphazardly on when we find them and when we don't, but it could solely be explained by the conductivity gradient. Still looking into that. Just thinking about the potential regulating mechanisms of this system, it seems like there is a strong affinity for the prairie stream fishes for the prairie stream reaches. What that exact mechanism is still might be a little bit elusive. What makes these shallow braided mobile channels with sand silt substrate seem to be prime habitat for these fishes? What fitness aspect is it satisfying? Why are they not as fit in some of these other reaches, say the eastern with more entrenched? Those specific mechanisms are interesting in thinking about, so maybe we can get at, pinpoint the regulating mechanism with that habitat of a prairie stream. And we also see the specific conductance gradient seems to be regulating. If I know what the conductivity level is in several of these reaches, I have high confidence that I can predict what fish you could expect to find there. So again, some more brainstorming, woefully incomplete at this point. We're still thinking through it, but just some obvious potential threats in this system. There's probably many others, but some things that we've observed and pulling together. Sodium chloride control projects have been ongoing for a long time in the upper Red River to reduce the amount of salt that reaches further downstream. There's a variety of ways that they try to control the amount of sodium chloride. Maybe it hasn't caused a shift in the fish community yet, but based on what I know, if we maybe clean up too much of the conductivity, you could expect a shift in the fish community. We do know in some of the drainages within the Upper Red River, water pollution is a current concern. Several of the smaller tributaries and so forth have some warnings associated with the amount of fecal coliform and other water pollution issues. In these systems, there's already several dams in place that help to fragment the habitat. If we see the genetics results, some of that fragmentation has not, at least so far, between the Wichita and the Peace River, hasn't affected the genetic diversity yet. But as more dams are come in place, how does it fragment the habitat? What does that mean for gene flow, for just the ecology of these fishes? Some question marks and things to think about as these projects move forward. And of course, in this arid region, dewatering or the in-stream flows are important for this system. I really don't have a handle on the amount of groundwater and how that affects the amount of surface water available in the area or pumping, but there are needs for water outside of the stream, and so those can have potential effects on the fish community because of fish that need the water, and then the amount of water also dictates the amount of specific conductance that is present there. So things to think about how these could threat and be a future threat to the current biota. Another thing that we did was use the SB3 work establishing of in-stream flow recommendations that has been used throughout the state. We use this type of approach to get some estimates, initial estimates on the base flow magnitude and the Peace River, Wichita River, using the same approach to estimate what would it look like to have subsistence flow, what number is that, and then also a way to, we think about high flows are important for this fish, but it provides us a common language on how to quantify those high flows based on magnitude, duration, timing. So it gives us a chance to provide context to think about how different levels of flows, going from a low magnitude but more frequent, all the way up to higher magnitude, low frequency, way to think about these flow pulses and how they can affect the fish community and the responses of these fish communities once these go through. In-stream flow recommendation is a stakeholder process, and this is not we're not recommending. It's just our first look at what would a recommendation somewhat look like for this system. So that we provided that in this report as well. So with that, I'll end with pictures of our Macrobopsis complex in Texas. Thank you.